Our last talk in this first section will be from Kevin Bird, entitled Anti-Racist Genomics, Responding to Scientific Racism in the 21st Century.
Great. I want to thank the organizers for inviting me. As a plant scientist by training, I'm always enthusiastic to facilitate cross-pollinating and hybridization. So I'm not going to be talking about my dissertation research, but instead sort of a second discipline I've walked into, studying scientific racism and using genomics to try to respond to kind of a burgeoning movement. And so there's been growing recognition of an expanding scientific racist movement with a confluence of advances in genomics and genetics and molecular biology and social media and the way that those end up reinforcing these communities. And there's some great work in the New York Times in addition to the AABA journal. And for those that don't know, this builds mostly off of genome-wide association studies. And so to quickly give everyone a crash course, a genome-wide association study is basically a souped up linear regression across the entire genome. So you have single nucleotide polymorphisms, abbreviated SNPs, and you correlate that with a phenotype. So in this plot, we're looking at height, and we're looking at the effect of a genetic variant on height. You can estimate the effect of that with the beta of the regression. And when you do that genome-wide, you get something that looks like this, the genome-wide aspect. This is a conceptually dubious application to years of education, which has been a hot button in terms of the scientific racist community. And this is built into the hereditarian hypothesis, which is a sort of masquerading scientific hypothesis with the ulterior political motive of naturalizing racial inequality and social, economic, and cultural outcomes. And the two main pillars of the hereditarian hypothesis are that European and African populations experience divergent natural selection on cognitive ability, and that these evolved genetic differences substantially explain racial gaps in intelligence and several sort of downstream traits. And by substantial, typically this means conservatively 50% or more. And to sort of spread this to create sort of the burgeoning movement, there have been several ways that a toolkit of science being misused and misapplied has been used to kind of grow the scientific racism. And so this comes from two main forms. The first is by co-opting mainstream research that typically takes an agnostic or a non-confrontational approach. It's sort of mainstream science. And we can see by an analysis on social media done by Jedediah Carlson and Kelly Harris that in this case, 10% of preprints on the server bioarchive that were analyzed had more than 5% of users commenting or sharing on social media that shared more than 2% of their follower networks, or their follower networks were similar with or had homophily with sort of the white supremacist reference panel that they used in the study. And so the plot on the side is basically graphing each dot representing a paper across several disciplines, and you can see the fraction of users that show great similarity in their followers with white supremacist networks. And these are heavily concentrated in animal behavior and cognition, evolutionary biology, genetics, genomics, typically papers that do very uncritical comparisons of racial or ancestral groups, or that take a very naive genetic determinist framework for interpreting and studying their project. In addition, there's actually several researchers that are publishing their own work, oftentimes in mainstream journals. And coincidentally, this research group has direct or indirect connections to the Pioneer Fund, the sort of mid-20th century organization founded on eugenics and white supremacy. And so this is some of the work that they're publishing, taking genome-wide association studies, comparing their frequency and correlating that to national IQ, doing it again 4 years later, showing plots like this, supposing to support the idea that genetics are the explanation for differences in national IQ. Other studies are looking at correlating European ancestry against IQ scores. Several have come out kind of here, showing plots like this, trying to prime people to assume that the genetics are being the driving factor of intelligence differences. And there's even cases where it's expanded off of comparisons of European and Africans or Black and white populations to explaining superior Jewish intelligence as perceived by the authors, again, using polygenic scores, genome-wide association studies. There have been lots of criticisms of this. Not surprisingly, the criticisms have been very indirect. They've been about the issues with interpreting genome-wide association studies, the methodological concerns, things like population stratification. There's a covariance between environmental and genetic gradients that produce inflated or spurious correlations. There are the fact that effect sizes don't translate across groups for several reasons related to the method of genome-wide association studies. Maybe there's dominance in epistasis that wasn't assumed. There's gene-environment interactions. There's differences in assortative mating, indirect effects, linkage disequilibrium, which is what genome-wide association studies use to sort of produce these correlations, may be different between populations. And so you miss a signal if you're looking at one, but not the other. And also just the aspect of evolution, genetic drift, genetic turnover, stabilizing selection even, can mean that populations will have roughly equivalent alleles, but they won't be the same between populations. And so focusing on one but not the other means you miss them. Now, the problem with this is these papers that are being published, I think, I see a connection to Bruno Latour's theater of the proof when he was talking about Louis Pasteur. Basically the idea that the perceptions and the optics and the persuasiveness of scientific results often have a stronger appeal than the actual merits of the science. And so here, these authors have produced several dozen papers proposing, showing positive evidence, showing plots graphed in very simple patterns where you can see a correlation and an increase. And what I think this does is it weakens the impact of the well-grounded conceptual criticisms that don't have the sharpness necessary to really pierce through the veneer that these scientific racist authors have made for themselves. And so what I have attempted to do is to take these criticisms a step further. And so my attempt at anti-racist genomics is to directly confront racist science on the ideological and the scientific grounds, incorporate critical perspectives on methodology and conceptual frameworks like genetic determinism, ground it in the most up-to-date sort of rigorous population genetic and evolutionary techniques, and in an attempt to try to break the theater of proof that appears to be very persuasive in online communities. And that culminated in this paper, which I ended up working on during my comprehensive exams, looking at polygenic selection between European and African populations and tests for divergent selection to put these claims into a more rigorous framework, to really explore them and to explore the ideological underpinnings of the hereditary hypothesis. And so again, these two pillars are going to be the main focal point of the paper. And so starting off with these claims about divergent selection. And so this relied basically off of publicly available genomic data from the 1,000 Genomes Project, in addition to the summary statistics from that education genome-wide association study. So for people who might be familiar with the Lewontin-Krakauer test, that basically is the underpinning of these methods. To just very quickly explain that, in yellow, we see basically the sort of genome-wide distribution of FST, a measure of genetic divergence. Most things are just going to be neutral. Their divergence is going to reflect neutral evolutionary divergence of populations. On the red, on the far side, these genetic loci show very high FST, which could be interpreted as a signal for selection. And so if we take this away from the original Lewontin-Krakauer framework of single genes and comparing the distribution of single genes, and instead take it into a polygenic framework, as the authors of this paper did, we can get basically the same idea, which is that blue distribution is the FST of SNPs not associated with height, and the red line is the FST of SNPs associated with height. And so when you see something far to the right, greater genetic divergence, that could be interpreted as evidence for natural selection. And so I did that using cognitive ability, educational attainment from the genome-wide association study. And by doing that, you find evidence very clearly against any sort of signals of natural selection. You either have something that suggests that evolution may be acting to make those genetic variants more similar in frequency than you would expect, or at the very least, they're evolving in expectation of neutral divergence. Now, this is a good and powerful framework. We get around some of these issues of effect size biases in genome-wide association studies by only looking at frequency and not worrying about effect size. But effect size is also important. Polygenic selection doesn't just act on effect size, it acts on the relationship between effect size and the frequency of a SNP. And so to kind of illustrate that, genetic drift would look like this. You have alleles changing frequency up and down between, in this case, French and Sardinian populations. And natural selection would instead look like this. So for example, we would say red lines represent SNPs that have an effect associated with lower stature, blue lines with higher stature. And there's a systematic relationship where the red lines associated with reduced stature are shifted to higher frequency in Sardinia, and the blue lines associated with greater stature are reduced in frequency in populations in Sardinia. And so this is sort of that covariance of effect size and allele frequency. And that would show you something like this, again, sort of using the same sort of bootstrap method. We're looking at things not associated with a trait, what that distribution looks like, what the differences between those populations look like. And in a genetic drift scenario, you would have roughly within the middle of the distribution. In a natural selection scenario, you'd have far outside the distribution. And here's sort of the key point. We're incorporating some criticism of genome-wide association studies come in, which is if you use a population level genome-wide association study where we have all these known biases and problems of interpreting them and applying them across populations, we see something that would look like natural selection. Now, if we sort of incorporate these criticisms and try a different approach, which is using effect sizes based off comparisons of siblings, which should produce a less biased estimate of effect size, getting around some of the issues that are plaguing genome-wide association studies in general, but not resolving everything, we actually see evidence not consistent with selection. And so by exploring those things and by keeping those criticisms in mind and applying different methods, we were able to get around a result that may have fostered more misunderstanding or would have fueled more misperceptions. There's some benefits of this. So we can, because of using within family designs and because by using these different approaches that don't rely on effect sizes, we have very well-grounded population genetic theory that finds no evidence of selection and suggests that these past results were driven by known confounds that people were saying were an issue, but were hard to really convince people when you have a theoretical criticism against someone's plot that they published in an index journal. I'm going to talk hopefully very quickly about the second part of this, which is trying to look at or get a picture of the possible role that genetic differences may explain. So this 50% claim is very pernicious. It's also mostly baseless. There's no genetic analysis that's being done. When there is analysis, it's highly inappropriate methods. And so the question is, if we can pierce through this sort of persistent speculation that is very vague and allows them lots of latitude to always change the terms of how they're trying to make the claim by using rigorous evolutionary frameworks, could we put more solid bounds that sort of chokes off the ability for this talking point to persist? And so by using some evolutionary genetics and doing an approximation of QST, which is the phenotypic equivalent of FST, since we know FST of trait-associated SNPs, we know or we can estimate different levels of heritability. And knowing that they're neutrally evolving, we can actually plug them into some very standard evolutionary genetic models. So one of the things that we can do is do the probability distribution of expected group differences based off of a highly idealized model of only additive effects, no gene environment interactions, all these things that we know aren't true. And still, when we make all these assumptions that should, if anything, benefit the hereditarian hypothesis, the majority of the distributions have extremely small, unimportant, negligible, potential sizes of genetic contribution to group differences. Again, sort of taking a simplistic idea of genetics and causation. Talking more about these model assumptions and violations, again, if anything, this makes the case, that would make the case stronger for hereditarians. But, and you would need careful investigation of the space of social environment, genetic context. And by kind of putting this out front and center, which I do in the paper, hopefully this chokes off the ability to say, someone did the same result, found a positive result, and so now we have solid evidence of the hereditary hypothesis. I'm going to skip this slide and talk about something hopefully more optimistic, which is using some of these more critical abilities to change genetics curriculum in high schools. And I'm going to highlight the work of Brian Donovan, who's just up the road in Boulder, working on creating genetics curriculum that teaches high school, middle school, college students genetics in a way that they understand the flaws with genetic essentialist arguments that sort of leads to this idea of discrete racial biological groups and discrete genetic causes of racial differences. And so Brian's been incorporating population genomics literature, even classical Lewontin FST results In addition to, we're trying to implement the results from my paper into curriculum to sort of start getting people to understand the dynamics of evolution, genetic differences, and through randomized control trials and like experimentally testing pedagogy and curriculum, it does have an effect of reducing students' racial prejudices and misperceptions about genetics. So hopefully this work can fuel more research like this and can fuel a better sort of educational grounding for students so that they're inoculated against these sort of misuses and misperceptions of genetics. All of this work would not be possible without people who helped me out with this or with NSF funding that has given me a lot of flexibility to do human genetics instead of plant science.